We're going to start off with Dr. Emily Coffey, who's from the University of Minnesota. And I was looking into your affiliation, and it's relatively long. So I just going to say you're from the College of Veterinary Medicine, and going to give us an awesome talk today about how there might be some synergy between awesome veterinary science and our field and how we can come together. So Dr. Coffey, I'll give it to you.
Perfect. All right, let me pull my slides up here. Yeah, don't worry about all of my weird background and stuff. You did great. That was a perfect introduction, so thank you. Okay, slides look okay and everything. I know we tested them before, so.
They look great.
Okay. Well, yeah, thank you for the introduction. And I want to start by saying just how much I love being a part of this group and appreciate how welcoming you have been. I know my work is a little bit different than most of yours, but really today we're gonna just kind of go through some of what we've learned from looking at the dog urobiome on the veterinary side of things. And my goals for today are first to just sort of introduce the dog as this potential model. I know that Dr. Hale has talked about this a little bit in the past, so there might be a little bit of overlap with things we've discussed. I am going to highlight some areas of our work. I actually didn't make this a very data-heavy presentation because really my main goal here is to just get people thinking about how we might be able to help you. So thinking about the goals that you all have within this group, ways we might be able to collaborate or work together to utilize these. I'm really focusing on dogs, but we have other animal models too. And, come up with ideas or, brainstorm a little bit. So that's really kind of how I wanted to frame this. And I'll start by just talking about the question of why dogs. And when I think about looking at the urobiome in dogs, I kind of think about this in two different ways. So one is, how can we use the dog as a translational model and learn things about the urobiome that might be applicable to human health and that sort of thing? But then on the other side of this, I also think that there are a lot of important One Health concepts about why canine healthcare in itself is actually quite relevant to all of us, in addition to them being able to answer questions about human health. So I'll start with this first one and thinking about the dog as a model. And I actually think there's a lot of different advantages. Some of you probably heard me talk about this before, so hopefully this isn't just exhausting to hear me talk about it again. But first and foremost, dogs actually suffer from a lot of the same urogenital disorders that people do, and they make really good natural animal models for a lot of these conditions. And I'll go through a small number of those later on as examples. When we think about them in comparison to more traditional animal models, like laboratory rodent models, things like that, there's additional advantages there. So for one thing, they have much larger bladder capacities, so we don't struggle with some of the challenges that investigators have that are working with mouse models. They have longer lifespans, pretty pronounced similarities in terms of immune system development and function. We know, so Dr. Hale had presented some of the really cool work she's done that shows the predominant taxa in the urobiome is actually quite similar between dogs and people. So we know that there's at least some parallels in urobiome composition. Similarly with the gut, we have some pretty good evidence showing that dogs are more similar in terms of gut microbiome composition to people than pigs or mice, which are, again, other common animal models. We also see really similar shifts in gut microbiome composition in response to dietary stimuli, medications, things like that. Another advantage is dogs are really inbred. So they've undergone a lot of inbreeding over the years. And as a consequence of that, we see relatively low genetic diversity within individual dog breeds. And so that can be helpful for us because, say, for instance, we're doing a case control study where we can breed match within that study population. It can really reduce some of the confounding variables of host genetics. Also, we know that there's all kinds of stuff that impacts microbiome composition in terms of the environment we live in, things we're exposed to. And dogs are living in our houses, drinking the same water, licking our face, sleeping in our beds. So they really share a lot of those same exposures and some of those less tangible things that are probably influencing some of this that are harder to control for in a laboratory setting. Another advantage is that we can really, I'm talking about pet dogs. So we have client-owned animals, and that can be advantageous in terms of the availability and some convenience factors for doing this type of work. We have a really, really busy teaching hospital that I work at where our clients are highly motivated and for the most part really enjoy having their pets participate in research. So that's really nice. And then like other animal models, we can control things like diet and other variables. So shifting gears a little bit to this other side of things in terms of canine healthcare itself, I want to emphasize that in vet med, we actually struggle with a lot of the same problems that human physicians are in terms of this UTI problem. So we see all these animals who have lower urinary tract signs and their cultures are negative. We struggle with the same diagnostic limitations. We definitely see a lot of antimicrobial prescribing for lower urinary tract signs. It's common. It's one of the most common reasons for antibiotic prescribing in cats and dogs. And I would say this sort of flow, this paradigm of, oh, there's a pathogen and now there's lower urinary tract signs and we're going to give antibiotics, is very much alive in the vet med world. And I think all of us in this group kind of appreciate that this is way too simplistic. This isn't really an accurate reflection of what's happening. And so I flip this around on people a lot. I'm like, well, we're prescribing antibiotics and we may or may not see an improvement in clinical signs, but probably what is happening is that we're at least promoting this environment for drug-resistant pathogens to develop. We're seeing shifts in what is actually this really rich ecological system that is probably not good in the long term for this animal. So we're struggling with this same sort of back and forth in the vet world too. And my argument here is that I think this applies or can affect human health, because coming back to this picture of some dog licking person's face, we have substantial evidence that we share microbes and microbial genes with our pets. And it makes sense given how we have come to cohabitate. So that includes antimicrobial resistant genes. And historically, when we think about antimicrobial stewardship efforts in the veterinary world, it's really focused on like food animal production and livestock. And there's been a lot more drive for stewardship efforts in terms of companion animal species. And we know that this can have downstream effects on environmental and human health as well. So better understanding the urobiome in dogs, what's happening, how we're prescribing antibiotics, what UTIs are, all of those same questions we deal with a lot in this group. Answering those questions in a veterinary setting does, I think, trickle down to human health as well. So just in terms of this One Health perspective, again, not only do I think that dogs could potentially be a good model and help in that way, but I also think that answering some of these canine healthcare questions can have positive effects on environmental and human health. All right, so I won't go overboard and pretend like dogs are just this perfect model. There is no perfect model. And there are really important disadvantages to consider as well. First off, they're dogs, right? They have different genetics. They're physiologically different in certain ways. There's ethical implications. Anytime you're using an animal model, they can't agree to these things, that sort of stuff. Another disadvantage is that the majority of our participants are spayed and neutered. Most dogs in the United States especially are spayed and neutered, and the exact impact of sex hormones on urobiome composition and things like that is not fully fleshed out. So that is one important difference we're often dealing with. There's also a lot of behavioral differences we have to account for, with a great example being grooming behaviors. So I strongly suspect we see far more contribution of oral microbes in dog urobiome composition relative to what you all see in people. And then you'll notice I had client-owned animals as both an advantage and a disadvantage. This certainly can be a disadvantage too. So it's logistically challenging. People have schedules. We have to rely on people for compliance if we control diets, to report things if there's clinical signs we're watching out for. I've certainly had people reach out to me saying, hey, I have this great idea for a study that I think will help us. And, you just have to have the dog like CT'd every 3 months or something. I'm like, yeah, people aren't going to let their dogs do that. So it's a double-edged sword, but again, clients can be both an advantage and a disadvantage. All right, so moving right along, I really tried to think about what to highlight here in terms of the work we're doing. And when I sat down to think about how our work might be able to contribute to the goals of this group, I decided rather than reinventing the wheel, I would kind of look at what you all have already put together with this word map we made early on in this group, and kind of use this as a roadmap to talk about things. So I'll start with this knowledge gap section, which is clearly a very monster section. But I'm just going to focus on a couple of areas in terms of stuff that we've done with looking at specific disease microbe associations, and then looking at some of the patient-specific variables that can impact urobiome composition. And as I mentioned, dogs, they get a lot of the same urogenital disorders that we see in people. So urolithiasis is a really big example, and I'm going to focus on that a bit because that's where my personal research is. So dogs do get kidney and bladder stones. Calcium oxalate is one of the most common stones. That's the one that I really study. Here you can see an example of a large calcium oxalate bladder stone in a dog. We also see really pronounced similarities with types of bladder neoplasia. We see a lot of neurogenic bladders due to various flavors of spinal cord injuries and traumas and things like that. And calcium oxalate stones they're quite common in dogs, and they exhibit a lot of very important parallels with the human condition. Here you can see just a couple of x-rays of a person and a dog, both with calcium oxalate kidney stones. And so my work has really focused on both the gut and the urinary microbiome in dogs that have these stones. And we've found a lot of similarities with what you all have found in people. So I put just one slide on the gut microbiome, even though I know we're focused on urinary here. But one thing that's really quite consistent between species is that we see lower microbial diversity in the gut microbiome of dogs with stones, just like people with stones. And specifically, we see pretty pronounced deficiencies in short-chain fatty acid producing bacteria. So this is really a particular focus of mine, looking at the relationships between short-chain fatty acids and stone formation. Again, similar to some of the work that's been done in people, the differences that we see just between groups in terms of overall microbiome composition are actually more pronounced in the urine than they are in the gut. And, whether these differences are contributing to stone formation. They're a consequence of the stones. They're a consequence of stone treatment. A lot of that is more challenging to tease through, and we're trying to do that. But we do tend to fairly consistently see that the urine of our stone formers is enriched with things like Pseudomonas, fusobacteria, and some Lachnoclostridium. So this is an area that this is kind of prelim data right here that we're delving into more. We do have some untargeted metabolomic profiles from these same dogs that were also strikingly different. Again, not super surprising. And we're doing some work now to try to figure out what's happening metabolically with these animals within the urine and how that relates to stone formation. Another kind of weird one that I threw in here in terms of this knowledge gaps category is we've done just a little bit of work looking at the impact of diet on urobiome composition. Obviously, diet is huge in shaping the gut microbiome, and we know a lot less about how it impacts urinary microbial communities. This was a small study with retrospective data. So we didn't prospectively control diets, but we had dogs that fell into these two different diet categories. And here I just put a quick taxa bar plot, but you can see some of the differences. And we did see reasonable differences between those two diet groups. Similarly, we also saw differences in dogs that had more diverse diets, meaning they ate a larger variety of things within a given day versus dogs who just ate their singular dog food and that was it. I don't want to oversell this. It was a small study. It was kind of pilot data. We're planning some better designed studies where we're prospectively controlling the diets they're on, and we're doing some crossover work and things like that. So I think that'll be a lot more informative. But I think what was interesting about this is it gave a little bit of support to the idea that we do see these downstream effects of diet on urobiome composition, which certainly that makes sense, and there's a lot of reasons why that might be, but it's an area that I think is a little bit understudied. All right, and then the next section is this experimental and technical needs. So the first thing I'll show you here is not very surprising, but just like in people, we have done work where, we collect urine samples using different techniques. So in dogs, the most common ways we get clinical urine samples are either through natural voiding. We'll collect these sort of clean midstream voided samples, or with what we call cystocentesis, what you all call suprapubic aspiration. You can see an image of what that looks like here. So that's typically how we're collecting urine. So we've done some work where we collect urine from the same dogs on the same day using these two techniques. And just like you all have found, we do see pretty important differences between those collection methods. When we look at the actual bacteria that are differentiating, we're probably getting a lot of urethral skin and genital microbes in the voided samples. So, again, parallels with what you all have found. And then the other area of experimental and technical needs I was going to highlight is weird, and I'll be very curious to see if anyone here has insight on this, but essentially, we were starting to get questions from people who did work in the water microbiome arena, where they're also working with these sort of large volume, low biomass liquid samples. And they would say, well, why are you pelleting the urine? Why aren't you using this sort of small pore vacuum filtration, which is what a lot of the water microbiome folk are doing? And so this is actually a small little study that Dr. Hale and I are working on together, where we took several different batches of urine and we aliquoted it, and we're pelleting them and filtering them at different volumes, and seeing if and how that impacts what our DNA yields are and what our sequencing results look like. So I was hoping I would have some data for that to show you today, but I don't yet. It should come back any day. But that was kind of a fun one. All right, I know we got a late start, so I'll wrap this last part up quickly. So cross-disciplinary research and clinical divide. I think it should be pretty obvious how we can be of help here. I think urobiome stuff is inherently interdisciplinary. We have, computational scientists, microbial ecologists, clinical urologists, all of these different groups kind of coming together. And this is another area where I think veterinarians can speak to some of the other biological systems that urobiome work can apply to, where I think it's just really neat for us to collaborate and sort of contrast and compare the types of things we see. I also, within this category, you had public engagement as one of the areas for improvement. And I present my work all the time to different types of audiences, scientific audiences, lay audiences. A lot of times I'm trying to, convince people that this matters to human health. Frankly, people love their dogs, and I don't think anything gets the public more excited than hearing about the types of stuff we're doing in dogs. It's like I see them glaze over when I start talking about human health sometimes. So I think there's an opportunity there to, when you demonstrate collaboration between these different fields and animal health being a part of it, I do think that can really get people excited and contribute to this sort of public engagement angle of that. And then lastly, bridging the clinical divide. I think that dogs can be really valuable when you have, say, an intervention that isn't quite ready to be tested in people yet, but is maybe at a stage where it would be valuable to test it in a natural animal model of a condition or a large animal model of a condition. This is, I think, where we see a lot of opportunities to really help. And just along these lines, this is kind of a busy slide, but when I take a step back and look at either work we're doing right now or work that we're planning to do or that we have on our wish list, I think a lot of it could apply to this idea of bridging that clinical divide. So I have all types of things planned in the stone research world that I won't get into, but in terms of intervention, looking at different dietary strategies, we can control relatively well dogs' diets, even with client-owned animals, different types of biotics. I have some work planned looking at downstream effects of FMT on urobiome. I think many of us at the Urobiome conference just heard this talk about BMTs, and that is something that we could explore. Looking at interactions with other biomes, colonization, looking at these other conditions, we see recurrent UTIs commonly, we see neurogenic bladders, all of these things where I think there's just a lot of opportunity there to potentially leverage this model as needed to help both species. And then I will end it by saying that I think the last advantage, if I haven't sort of beat this to death already, is that you in this group have access to two very enthusiastic and collaborative canine urobiome researchers with myself and Dr. Hale. Hopefully she doesn't mind that I'm throwing her name out there so much, but both of us, we love this stuff. We're happy to chat. Sometimes I've had people reach out and it was something where we were able to make it happen, and sometimes we decided a dog wasn't the best model, so we're happy to chat about that stuff and see. So that's really what I was hoping to do here, was just sort of get people thinking and remember me or us if you have ideas where you think we can be helpful, because happy to do it. So, or at least chat about things.